In this video, we're going to go over how to make mutations in a protein sequence and go all the way through cloning them into different cell types. So to start out with, we're going to look at this DNA sequence, and we're going to be looking specifically at this protein CPS6. So I want to make a mutation at the cysteine site. So what I'm going to do is actually copy out the sequence that comes before the cysteine. So here we go. We're going to paste it into a text file. And then we'll go ahead and find an alanine to put in there because that's the mutation I want to make. And then copy everything that comes after the cysteine as well to the end of the protein. So once we have this all in our doc, we can make a new DNA file and paste that in there. And then make sure that we label it so that we know what this DNA sequence is going to be. Then we can take part of this sequence and search for it. And then we can localize that mutation that we made. So I am adding a feature right here just so that I remember where I made the mutation. And then from there, what we're going to be doing is making primers. So I go about 7 base pairs upstream of it, and then we're going to have 30 base pairs total for our forward primer. So we're going to go ahead and add that primer and label it. And then we want 15 base pairs of overlap. So we go back 15 base pairs and then select a bottom primer and add that label as well. So now we have our 2 different primers with 15 base pairs of overlap. We can look at the primers on the primer tab, and we'll be able to use those when we want to order them. So I used IDT in order to order my DNA oligos. So I'm going to go ahead and type that stuff in here. We're going to label them so that when they come in, we know exactly what they were. And then the nice thing about SnapGene is we can go back in there to the primer tab and copy that sequence into IDT directly and do that for the reverse primer as well. And then go ahead and just complete the order. I always make sure to double check everything to make sure it looks good and then go ahead and check out. So once it gets in, we're going to go ahead and take these, and IDT does it where they label the nanomolars that come in the tube. And so what we're going to do is multiply the nanomoles by 10, and that's how many microliters, 0.1x TE we're going to add to each of the tubes. Once the tubes are ready to go with that, we're going to put them on 50 degrees C for 5 minutes, and then we're going to make a 1 to 10 dilution into water. Those primers can then be stored at -20 C until they're ready for use. So we want to make this mutation happen in our protein. So what we're going to be doing is doing a PCR at this point. So we have a PCR tube that we're going to label real quick, and then we're going to add Hi-Fi, which is an infusion mix that we use in our lab, and we're going to do 12.5 microliters of that, 9.6 microliters of water, 1.4 microliters of our standard plasmid, and then 0.5 microliters of each of the primers. Those get loaded into the thermocycler, and then they're going to run in a normal PCR reaction, with the only difference being the 72C step, which we're going to increase to 45 seconds. And a general rule of thumb is that you do about 5 seconds per 1,000 base pairs, and my plasmid is about 9,000 base pairs. The next thing that we're going to do is make our Agarose Gel. So we're going to mix 0.25 grams of agarose with 25 mils of 0.5x TAE, and then we're going to place that in the microwave for about 30 seconds and then check on it constantly. So when it starts to bubble, we're going to take it out and stir it a little bit, and then after it looks clear, we're going to add 3 microliters of CyberSafe and pour that into the gel well. So once the gel is set, after about 30 minutes, we're going to add an additional buffer solution of 0.5x TAE till it fills the top of the gel, and then we're going to go ahead and load our sample. So I pipette 1 microliter of the loading dye onto a piece of parafilm, and then add 10 microliters of the sample to that mixture, and then load that, and then we're going to let that run for 30 minutes at 100 volts. Once that's complete, we're going to go ahead and image that. So we take it to the imager and load it in for the CyberSafe stain. And once that's done, we can look at it. And what you can see here is the gel with the band size that we're looking for. So that's great. Now we're going to do Clon Enhancer on it. So we add 1 microliter of Clon Enhancer, 7 microliters of water, and 2 microliters of our sample. And that's going to go at 37C for 15 minutes and then 50C for another 15 minutes. Once that's complete, we're going to go ahead and transform our DH5 alpha cells So we're going to mix 90 microliters of our DH5 alpha cells with 10 microliters of our clone enhanced material, and then we're going to let that sit on ice for about 20 minutes. Once that's done, we're going to heat shock it at 42°C for 35 seconds, and then add about 1 ml of media to that solution. Then we're going to take that over to a incubator and let that shake for about an hour. While that culture is shaking, we're going to go ahead and grab a media plate that has kanamycin in it, because our plasmid has a kanamycin resistance in it, and we want to challenge the cells against that. After the hour is complete, we're going to take about 100 microliters of that cell solution and dispense that onto our plate, and then using glass beads, we're going to spread it around, and then we'll load that into the incubator so that that can grow overnight. The next day, after you see that it's grown, we are going to make overnight solutions with that. So what we're going to do here is add 4 mils of media to each of these tubes, and then we're going to add our kanamycin again, so that's going to be 12.5 microliters of kanamycin, and then we're going to carefully pick individual colonies out of this and set those in the incubator overnight so that they can grow up, and then the next day we're going to come and spin them down. So we're going to look for the cell pellet, and that's what we're actually going to extract the DNA from. So once they're spun down, you're going to go ahead and pour off the media, and then I put these on a piece of paper just to remove any extra media. Then we're going to resuspend this solution and place that in a tube. This is a Monarch test kit. It's just something that we use in our lab. There's plenty of other kits that you can use, but this is what I typically use. So now you can see I'm adding a lysis solution, so that breaks up the cells, and then there's a quenching buffer which stops the lysis from happening. So now we're going to load it into a centrifuge and let it spin down, and then you'll get the cell pellet that you can see at the bottom, which is going to be the membrane. So we're going to remove the solution and put it into a spin column and load those spin columns back in, let that flow through the column, and then we'll go ahead and remove what flowed through, and then add our wash number 1, and then add our wash number 2. And then I add 50 microliters of the elution buffer to each of these and allow them to spin down. So here I am prepping to send off for sequencing, so I have PCR strips, and I'm going to add 2.5 microliters of water and 2.5 microliters of my sequencing primer made to the same concentration that we talked about earlier, and then add 10 microliters of each of the mini prep solutions. So once we get the sequence back saying that the reaction worked, we're going to go ahead and transform those cells. So we can take EL21 or codon plus cells, which are used for expressing protein, and we're going to add about 5 microliters of the mini prep solution to 90 microliters of our codon plus cells, and then we're going to do the same transformation reaction that we did previously with the DH5 alpha cells. And then for this plate, we're going to make sure that we add chloramphenicol to the plate, because the codon plus cells have a chloramphenicol resistance to them. And then the next day, after this is grown up, we're again going to make overnight solutions, and this one is going to be used to make a glycerol stock so that we can just pull from this in the future and use this to express our proteins. So we're going to let this grow overnight, and then we're going to add 400 microliters of 75% glycerol, and then 600 microliters of our cell solution, and then we can store this at -80 until it is ready to use.